be working, like this one is now. And, you know, we have a couple minutes to ask Dr. Studer about some of the themes he talked about, or specific questions about, you know, his own experience in Parkinson's. Okay.
First, thank you. That really was a perfect introduction. It's much appreciated. One piece that you mentioned had to do with the use of fetal tissue and fetal cells. Obviously, in the United States right now, we're in a difficult time with that. Can you just say a few words about exactly where they are most essential and what possible substitutes we could use, or if there are areas where we're simply going to be stopped until that can be eased up a little bit?
Sure. I mean, I think the areas where they were most essential for us is twofold. On the one hand, just simply to tell us how nature makes a given cell. So do have the standard, and that's, for many years, has been unfortunately kind of ignored. So people just use the fetal tissue and maybe try to transplant it. But now, with new techniques, there's a big effort across the globe. And I mentioned this One Cell paper just coming out last week, where systematically every cell is profiled in a way that we're going to have a roadmap of what really a normal cell looks at any stage. I think that's very, very important to have this reference point. That is really, I don't know how else you would get that. No, that I think is absolutely essential. So we're going to need fetal tissue for doing that. And the other part in the Parkinson's field is, again, to learn from the successes and the misses in the fetal tissue trial. Obviously, in Parkinson's, we don't have time to discuss that, but there were a lot of problems that also came up with fetal grafting. And we learned a lot with regard to patient selection, with regard to areas where things basically didn't work very well with fetal tissue and where they did work very well. So it had this kind of learning curve for the last 20 years that is actually very instructive for the stem cell-based work now to move forward.
Theodore Renault from Altius. Thank you for a very informative presentation. I had a question about the GMP grade cells that you have made and banked. I'm wondering if you could share a little bit with respect to your plans for early stage clinical development, inclusion criteria, route of administration, cell dose, anything along those lines.
Sure, I mean, that's obviously a very big theme, and I'm happy to discuss in much more detail, but I mean, there's a very important point. So we had to define facts for inclusion criteria and exclusion criteria, particularly factors that we think are obviously critical for function and safety. And so there's a very range of specific dopaminergic markers that are included in quantitation. When I showed you FOXA2, the key marker we needed to, but we typically have 99% of that marker expressed, so this would be all coming from that ventral midbrain domain. But you need to exclude markers, not only pluripotency marker, that would be worried that you would have any contaminating cells, which we seem to have 0, but also other neural cells that need to be excluded, very important. And then with regard to potency, that was again actually an area which fetal tissue came in very, very handy, because it turns out that some of those fetal grafts, dopamine grafts in patients where they seem to have worked reasonably well, again, which ones work, which ones not, we can discuss later. There were about 100,000 surviving dopamine cells in autopsies. You actually have autopsy material. And so we actually use that as a dosing criteria for our cells, where we flank that range that we know is efficacious with fetal cells. And again, I'm happy to talk much more in detail on the various factors.
I wanted to start a discussion. Kapil Bharti from National Eye Institute, NIH. I wanted to start a discussion on regenerative capacity versus paracrine effect of the transplant. I think it's not only scientifically relevant, it's also important when we reach phase 3. So how does one really prove beyond doubt that the cells that you transplanted are really integrating and the effect that you see is because of a regenerative capacity of cells? And you touched briefly on that, but can you expand a little bit more on that?
Yes, again, there are these kind of techniques where you can just simply functionally inactivate your cells. With neurons, it's kind of easy. That's what I showed, optogenetics, or there's also chemogenetic techniques where you can do that. And again, we did physiological studies combined with it, where you actually record from the cells that you know should receive the signal, should get dopamine modulated. And so we get pretty close to an understanding of how these cells function and integrate, and how long it takes them to get to that stage. Now, what it doesn't address is whether on top of that, they might have paracrine effects. What we hope in this case is that the fact that they do connect leads also to basically preservation of the synapse. That's something, again, there is some evidence for that in animal models, secreting facts like BDNF, that usually when you lose dopamine neurons over time, you also lose the dopamine receptive cells at late stage of the disease. So we hope that those paracrine effects will actually preserve that. So you not only have the function of the cell, but via paracrine effect also the preservation of the circuit But again, so there is, for each disease, I think you have to have your strategy. You can have knockout cells, that have the part that's missing that you think is essential for your cell. So there's, I think, a lot of ideas how you can do that, but very, very, very few studies that actually have done it.
Thank you.
J. Siegel, Johnson & Johnson in the forum. Thank you for your presentation and for a rather broad and I think outstanding overview of many of the challenges you faced. One you spoke of was you emphasized the importance of making decisions in the face of uncertainty. I think you were talking about, for example, purity or choice of cell line, and you mentioned regulatory uncertainty. And I wonder if you could provide us information about the source of that regulatory uncertainty, because I know the FDA tries to provide guidance, but there's rapidly evolving science, and were there issues of not being able to reach regulators in a timely way, not having access, or did they just not know the answers yet because it's too new, or where is there regulatory uncertainty where you might be able to get more assistance?
Yeah, I mean, I think it's a good question, and obviously, I think, again, the FDA tries to be clearly very helpful, and I think tends to be accessible, coming to meetings and discussions and so on. But there is just, I think this uncertainty has in part to do with the fact that that they themselves don't know yet how to regulate the product, because there has been no path yet. And that's the famous example when Geron first came up with the trial. They had whatever it was, 27,000 pages that they submitted to their IND documentation, because so many things that they didn't know, and they came up with new things that had to be tested. I think that uncertainty is going to go down over time, once more and more products come through the pipeline. But I think for us, the big one is really the cell line choice. So we and other groups actually pursue the so-called legacy lines, like H9, that were grown on feeders at some point, were made GMP-like, requalified. And clearly they seem to be FDA allowed, so they go into early stage trial. But the question is, can they become a clinical product? And there we cannot really get a very clear answer, because the question is, I guess it depends, and things might change by then, and we learn more what happened in those early trials. And so I think that makes a big change, because if you have to start all over again later with a new cell line, it's nearly you have to go through all the studies again. And so that kind of needs a leap of faith that you think it's going to be right, and maybe some backup strategies behind. But that gives definitely certain uncertainty. So I think a lot of the uncertainty is just simply because the field is still uncertain on what's the best way forward.
So Lorenz, thank you. This is Lucy Brain, Chief Scientist for the ALS Association. And it's probably more a comment and an observation, and perhaps we'll discuss it more during the day, but you highlighted some challenges. I think one of the big challenges, for example, in the ALS space, we have some clinical trials going on, and to get funding support for phase 1, phase 2, CIRM's been fantastic there, NIH as well. But then you have to get to the phase 3 definitive trial, and small companies are obviously struggling. We can't afford to pay for it. And we're starting to see that in some ways these companies are starting to charge for the procedure before it's gone into phase 3. And I don't want to name names, but we're dealing with this very much at the moment. So I don't think you have a clean answer for that, but is there lessons learned from other disease areas that can help us? And how do you see this moving forward?
Yeah, no, I think that's, again, a very iffy issue with regard to having, again, patients pay for something that's really not approved. And we just discussed that actually in great detail in the ISSCR guidelines, where we discussed this issue in great detail, in which cases that might be an adequate case. And in ALS, obviously, you can think it's a very end-stage disease, no other options. But on the other hand, I guess the rationale for some of the therapies is also not really that strong that you can say it's an absolute no-brainer, you have to do that. So it gets into a very gray zone. And I think at the end, I think the approach needs to speak for itself to actually have a chance long-term. I don't think it's a solution to go forward like that, have patients pay for that on a long-term basis. So I think you need to eventually, and again, it needs a change also in the investment field. Now it needs actually the hedge funds and other companies, there are very, very few right now that actually go into this field exactly because of the long timeline. But I think there are some areas, at least, where we start to interact with people that realize the potential and that might be going in for the long term. And I always think of the example of CAR T cells. Now, CAR T cells, we have to see how it's going to go long term, but there has been obviously an explosive growth financially from a therapy with those cells where initially people could not get any funding, exactly were at the same value of death. And maybe suddenly when the first patients came along, then suddenly these companies had $200-300 million money just to start up, and now they are valued at $3 billion. So it kind of exploded very, very dramatically. And it's quite possible now that in regenerative medicine, such a step is going to happen as well. And then once that happens, I think that might be easier for things that have chance of success to really also get that next level of funding, which again, as you said, probably can no longer be a patient foundation.
Yeah. Thank you very much.